Nice talk, Michael.
Good stuff.
Thank you too. Yeah, it was all good.
Everything.
You too, punk guys.
All right.
You too, Marv.
All right, welcome back. So this is the last part of our discovery track, and this is our NextGen session panel discussion where we can have some live Q&A, session. We didn't get a whole lot of Q&A going on during the talks, so maybe that's a good time. Maybe our clients saved all of their questions for this particular session. We're going to be focusing really on pushing the boundaries beyond what's already done out there, and there are a lot of areas that can benefit from pushing these boundaries. So I'd like to see if there are any suggestions. I'd like to see the attendees that we have here, and if our attendees would be kind enough to contribute to the discussion panel, that would be great. We've got a few topics that we've pre-selected to sort of seed the floor with some sort of hot topics of the day. But as we advance in this hour, then we will be drawing topics from the Q&A pane. So and it seems that there are really different areas where we can push boundaries of science. I think some of the challenges that we see and that our clients see right now is sort of pushing the boundaries of chemistry space, and in essence, pushing the boundaries of the domain of applicability of our models. I mean, essentially, that's what pushing the boundaries of chemistry space really means. And there's some parts of chemistry space that are kind of difficult to tackle, and they can be tackled in different ways. We get a fair number of clients who are involved in PROTAC ligands. Also, a fair number of our clients are involved in macrocycles. So, like two parts of chemistry space. We do have some of our models do cover that. Not all of them do. I think it's fair to say. And there are different approaches that can be taken to tackle expansion of chemistry space. I mean, it could be through federated learning. Is certainly one big avenue that's already been discussed out there. But it can be more targeted. For example, in PROTAC, we could approach selectively our clients about having data collaborations where they could, of course, with the proper confidentiality in place, share data with us, and again give us access to these molecules and this data, where we could expand our models, maybe specifically for PROTAC ligands or specifically for macrocycles. So there's different different possible avenues where we could go here, and so I'm going to open the panel to our experts, and I think we can go around the table and see, which of these approaches can best contribute, to that expansion and to that enhanced applicability of those models. So maybe we can again go around the table and get everybody's sort of sense of what would be the most likely direction or the most productive direction for us to go in. Who wants to start? David, you want to say a few words about this as leader of the group, leader of the R&D, at Predictor R&D?
Let's see, is my microphone on?
Yeah, you are on.
Okay. I'll leave it to some of the others to provide their input. The one thing I'm not sure we'll get a chance to talk about today, but Michael alluded in his talk, which was on AIDD, to a feature that we'll be adding to the next release of ADMET Predictor, which is the generation of 3D structures. Now that's something that we get asked about from time to time, do our predictive models take 3D information into account? And of course, we've found over time that we can get a lot of mileage out of 2D. But I think the question remains is can we utilize a 3D structure to improve some of our predictive models, particularly those that are related to physicochemical properties and potentially to metabolism. I don't know that we have anything on that front to announce today. This is something that we're going to do gradually. For this release, we'll have 3D structure generation. But I think over time, by integrating some 3D information, we may be able to get some improvement in our models and, perhaps, as you said, tackle some areas of chemical space that up to now, we haven't been able to. So I think with that, I think we have experts in model building here on the panel that certainly have more expertise than me. So see if anyone else wants to chime in.
Yeah. Well, I mean, maybe one thing to see it a little bit further, I think, I think it's highly model dependent. And maybe there's some areas like, say, pKa, for example, where maybe 3D is it, I mean, does it really, like that steric hindrance really make a huge difference? I mean, given the size of a proton, which is really, really small, does it make a big difference on ionization constants? I'll let Robert or Michael comment on that one.
Yes, it does.
Yeah.
The example are so-called proton sponges. These, the simplest one is doubly substituted naphthalene with 2 tertiary amines on nearby on both rings. The 2 amines hold the proton, and the 3D structure arrangement of these atoms is crucial. Our 2D model does not capture that aspect. So the predicted pK value is lower than the measured one. Yeah. So yeah, 3D could benefit pK prediction somewhat.
Yeah. Somewhat.
Yeah.
Again, I mean, it's about kind of the sensitivity of 3D to. I'm thinking of permeability, for example, versus macrocycles. I think it's pretty obvious to a lot of people that the conformation of something like cyclosporine A has a huge influence on its permeability, and that's
right. So so macrocycles, well, just anything going through a membrane, the fewer hydrogen bonds it makes with the molecules within the membrane, the faster the permeability will be. So one thing these macrocycles can do is when they get in the lipophilic part of the membrane is they fold all their hydrogen bonds internally and form internal hydrogen bonds rather than with the membrane itself. And so that would increase the permeability. And so that's a feature of the 3D coordinates. For pKa, this Rambaugh article I was talking about with the base 1 inhibitors. So, that ring is mostly saturated, and the substituents in the positions on that ring, if the fluorine is up in one case, that'll affect the pKa of the amidine ring, and if it's down, it will change it. So the distance of the amidine functionality to the fluorine ring really does affect the pKa of that, and it you can see from that publication that different stereoisomers resulted in different pKa's for that compound. So that's one thing. Maybe Robert could potentially talk a little bit about that. Maybe.
I have a quick comment on that. Actually, I agree. Having 3D structure is definitely very useful, but how are we going to make sure that the 3D conformation we are looking at or we are dealing with is the correct conformation? As Michael just mentioned, That's in case of macrocyclic or even the cyclic peptides, there are internal hydrogen bonds, and that helps in increasing the permeability of the compounds when it is passing through the cell membrane, and then once it is on the other side of the cell membrane, then it rearranges itself to make it more soluble in the cellular fluid kind. But again, this is where I was bringing in the point that even if we are looking at 3D structures, we have to be like the user has to be aware that there are certain limitations of using the 3D structures. We cannot be very like the user cannot be very sure that the 3D structure what he or she is dealing with, they are the correct 3D structures in terms of 3D conformations.
I don't know if there's been such a thing as the correct, because even the crystal structure is still a kind of a picture, so or a snapshot,
static
picture, exactly. But Fiofio has a lot of experience in the work that was done in the group of Denis Fourche, I think, at NC State, that did a lot of conformational averaging of properties, if I remember correctly. So, Fiofio, is that something you want to comment on?
Yeah, so what I was working on at the time was working with like PKS enumerator, where I was designing a lot of macrocycles. So I had a chance to play around and work on different conformations of macrocycles, and I also did a lot of reading during that time. The problem with especially for macrocycles, is that if you change even like a small functional part anywhere within the ring, it can significantly affect the conformation of the entire ring, which is really problematic. And it is still a challenging issue that needs to be resolved. So when I was working on that, it was still kind of like a developing area. There was really like no good solution on that. I know that there are other programs out there that try to tackle this conformation sampling very differently. Sometimes they split the ring into halves, and then they try to generate different conformations separately and join them back together. But even that approach hasn't been like proven successful in terms of trying to predict the good conformation. There is still a lot of research, especially that needs to go into the macrocycle conformation.
Maybe David could comment on how he generates 3D coordinates for macrocycles in this new program, and different conformations too.
Yeah. Okay. Michael's going to put me on the spot here. Yeah. So just to clarify, so this next release of Predictor is scheduled for end of April, I believe. So it's coming up quickly. I think we'll have more information to provide at that time. We haven't written any documentation or sort of gotten ourselves prepared to, introduce some of the new features yet. But macrocycles are hard. Anyone who has worked on 3D structure generation knows that. I don't want to say too much right now about the algorithms themselves, but the 3D structure generator is not based on fragments. So I'll just throw that out there right away, as some are, some very good ones are. This one is not. It does, however, make use of distance geometry for difficult ring systems. So caged ring systems, bridge systems like morphine, and macrocycles. It does have the ability to generate conformational ensembles, including for macrocycles, and there are a number of settings the user has control over to affect the number of conformers, the similarity between the different conformers, and so on.
And the energy, that's a big part too.
Yeah. So there's an implementation of the MMFF94S energy function that is used in various parts of the structure generation algorithm. So you have some control over the total energy difference between the lowest energy conformer and the highest energy conformer, and so on. So we'll have a full presentation on this in less than 2 months, I think. So I can just say stay tuned, and we will be interested in getting feedback on the quality of the 3D structure generator itself, and of course we're starting to think about how to apply this and actually use it internally for things like property prediction. That's probably going to be a long process, and we're interested in in working with some of our clients on that as well.
Mm-hmm, mm-hmm. Yeah, good stuff. All right. Anything else on that particular topic?
I'll just say that it's very what David's programmed is very rigorous and fast. So I've submitted some complicated ring structures to our new ADMET Predictor 3D generator, and it generated them very quickly, whereas other programs didn't generate the structure at all after 20, 30 minutes even. So.
I will like to second that as well. Like there were few ring systems which I was looking at the 3D structures, and I compared few 3D generator softwares which are available in public domain, and the ring system coordinates with our new program seem to be really promising compared to the ones which are available in the public domain.
Yeah. Good stuff. So yeah, I mean, sort of coming back to one of the topics that I mentioned on this again expansion of chemistry space is access to data. That is a very important thing for us. Like everybody else, we have access to the public data. I think pretty much all the companies are on the same page of that one, but also realize the limitations of what's out there, and that on some occasions our clients can work on parts of chemistry space that are very specific, and if we don't have data to accommodate that particular space, then there are a few options that are available to them. One of them, of course, is to either build a local model or expand our models using their data. But one of the scenarios we like a lot, and we've been very fortunate to reach that status with several of our customers, and Bayer in particular, is to get access to their data, and about 2/3 of the data in the pKa model, if my memory serves me correctly, comes from that collaboration. So, I mean, whereas if everybody is working with 10,000 ionization points, we're working with 30,000, and with that in hand, we can build a better model, and have a domain of applicability that is much larger. Now, that is the kind of scenario we'd like to repeat, and that's really an open invitation to our clients, that if they're working in an area where they think the community would really benefit, and areas like PROTAC are certainly one of them, microcycles are another one, but we really value a lot access to this data, and of course we'll be talking about various things about the business terms and so forth. That's not the forum for that, but the point is that we are very interested in working with our clients in securing specific data sets that will help them, of course, but also the community at large. And of course, we guarantee the confidentiality of the data, and we do actually purposely make certain aspects of our software that cannot be reverse engineered in terms of trying to guess what's in the training set. So we do safeguard that data in multiple ways. So I just want to put it out there that it is something that we not only that we do, but we've done in multiple instances, and with, again, companies who are as large as Bayer. So rest assured that interest is quite alive. So yeah.
So I think that's the cue into the a little discussion about federated learning. Is that correct, Eric?
Well, we could talk about that, certainly.
Okay.
That's another avenue. Robert, I think you may have seen more of this, federated learning, and maybe expose, all of us a little bit more about this new avenue that's opened up, a new format for working.
That's correct. I have just shared my screen. Time and again, over decades of experience as a simulations plus scientist, I have encountered the same problem. Our customers often complain, well, our compounds show as an out of scope in some of our models. Okay, well, sure, you have a very specific chemical space that is tailored to your discovery and development project, which is not or may not be quite overlapping with the public space. Public means the published chemical structures in the literature. So the ideal way to solve this problem is for you to share your compounds with us and your data with us, so we can append it to our training set and build a model that covers your chemical space. And that's what we have exactly done with Bayer data, and build a PKA model that joins both company and public data. And the model is widely successful. The other why didn't we why didn't we do it with everybody? Well, time and again, scientists are very much interested in sharing the data. The company lawyers are not. So that's an obstacle that is quite difficult to overcome. The recent developments in machine learning are now having their own Wikipedia page. It's called federated learning or collaborative learning. It's a machine learning technique that trains an algorithm across multiple decentralized edge devices or servers, holding local data samples without exchanging them. So in other words, in practical applications, several companies can come together and decide, okay, let's build a global model of, let's say, volume of distribution that will benefit everybody without sharing our own data. There are several scenarios in which that can work, and you can take a look at this page and read more about it. One is the centralized federated learning, where there is a central entity that is trusted by everybody, that essentially develops a system that builds such a model. An example of actually functioning entity like that is a Melody consortium. You can take a look at this. This is a centralized way of building predictive models. It's very elaborate and requires essentially everybody to be synchronized in the same time. Because the central server will build models using itself as a director and company servers as workers. So everybody has to be synchronized in the same time and so on. So it may not be a convenient way to do that. We discussed an alternative idea to federated learning. And let me show the basic tenets of it. You have seen this slide several times already, how we built our ANE models. But the important part is that we end up with an ensemble of models. Okay, and this ensemble is, of course, built on the same computer, or the same server if you prefer. The default number of networks is 33. Well, there is no obstacle in having our customers use our ADMET modeler software to build parts of such an ensemble using their data. Okay, the data doesn't have to be shared at all. Okay, the members of the ensemble could be generated at individual companies without sharing the data. All we would do as the supervising entity was to collect the resulting neural networks and put them in one large super ensemble. And I'm entertaining this idea. We actually have allocated resources for a future test of this idea. And I would be very grateful if, let's say, Eric put up a poll that would ask whether companies are interested in something like that, or how much interest is there. And I would like to know the answer. Also, please use questions and answers tab to ask the relevant question there. That's it. I will give microphone back to Eric.
Yeah, I was just typing it, typing the poll question, but yeah, I mean, this is certainly another possible avenue to to that expansion of chemistry space, and again, getting synchronized with the different types of chemistries that are happening, at our clients' sites. So that's certainly a very good one. Now, there is yet another topic that we could go into. There is one one in the Q&A. Okay, I need to bring up the challenge. Oh, okay. Then yeah, so another topic that's been hinted, well, more than hinted, it's been presented this morning, is by Marvin, dealing with tautomers and how to pick the right tautomer, essentially, from a set of possible ones. Now, because the output is so sensitive, I think there's probably a question on a lot of people's lips about the ability to potentially make the predictions tautomer independent. So that is a topic that some of us have had some experience with, and maybe likely something that would be of high interest to our clients, because it removes one variable clearly from the prediction. So maybe that's something we could cover a little bit as part of this panel. Robert, did you want to start sharing a little bit of thoughts on that? I think you asked me to include it on the panel, so I did.
Yeah, sure. No problem. You're talking about tautomer independent modeling, correct?
That's what I'm talking about.
Yeah. It turns out that what Marvin has developed in terms of predicting dominant tautomer is one aspect. In another is the fact that the model outcomes do depend on the input tautomer. And this has been a problem raised mainly by Yvonne Martin back in early 2000s, as well as Hugo Cubini. They truly wanted somebody to make that dependence go away. So, in other words, regardless of which tautomer is submitted, we would like to, or they would like the computational chemistry outcomes to be identical. Okay. And we had started actually working on this problem back in 2014. We worked it for about a year, and unfortunately, at the time, we had to stop because there are more pressing things to take care of, more pressing projects and so on. So we came back to it last year. Let me just share my presentation that I have developed. Sorry, technical problems. Let me share my screen. That's correct. Yeah. So that's an old slideshow that I have presented back in 2015 in Denver. The problem of QSIR, the methodology of QSIR modeling is very well known. We have molecule, we call it as descriptors, and then generate models that predict properties and so on. And yeah, there's an elephant in the room that nobody wants to talk about, is that the chemical structure that you draw and see on web pages dedicated to various drugs is not what really the living organisms see. Okay. This is not the structure that is actually doing its action in the biological sense. It's not the structure that is responsible for its efficacy, because in water, this chemical compound undergoes a lot of processes. Okay, there's an ionization for once. Okay, there's tautomerization, so it may exist as different tautomers. There's aggregation, micellization, complexation, and all of these. A collection of various and distinct molecular structures. That's what is responsible for the biological action of the drug. Okay, and presenting one particular tautomer, okay, to the programs like Admet Predictor results in different outcomes, okay, because there are different descriptors, as you can see, for different tautomers of the same compound. Different descriptors result in different models and different model outcomes. There's a PK, for example, okay, well, I have different results for PK, I have different results for log P and everything else. So this is just a reminder that tautomers are nothing but protic tautomers are nothing but protonation microstates. And this is the series of slides dedicated to that particular aspect. Nevertheless, okay, I'm going to skip that to make the long story short. We have succeeded in actually unifying the structural representation of a chemical compound, so that regardless of which tautomer you input, you get the same results. So you can see the before and after the situation. For this particular molecule, it's easy to create such a unique representation, so that regardless of which input tautomers you have exactly the same PK predictions. That was the state in 2015. There are a lot of other problems to be solved, which I took on starting last year. And now I can say that I don't have, unfortunately, slides that we illustrated. But now I can say that with about 99.4 accuracy, we have conquered this problem. And I'm working on rebuilding a PK model using this unique representation. How much interest is in this? I hope there will be a poll. If not, there may be some questions asked. So yes, this is the project that is ongoing, and when it's finished, I'll be happy to do presentations and posters and actually share the results with our customers. That's all from me.
Okay, I'm going to launch the poll tautomer independent modeling answer 1, high, medium, Medium, low, low, and save. Let's see if I can run this as a poll. What is your level of interest in tautomer independent modeling? Launch poll. There we go. Let's see if that works. Did that launch? Oh,
there it goes.
There we go. So let's let that run and for about 30 seconds like we've done before. Okay, let me retrieve the poll and see what the so it's about 50% high, 25% medium, and 25% low. So it's fair to say that 75% of our customers thinks it's anywhere from moderately to highly important, and I think that represents pretty fairly what we know to be true. That it is something that's on a lot of people's mind. The elephant in the room, as Robert mentioned, that that most of the algorithms, just because of the descriptors that they involve, would be dependent. The output would be dependent on the tautomer input. So that is obviously a potential problem. So removing that variability, I think, would certainly help a lot of people. I think that's a fair conclusion. All right. Well, we have about 20 minutes left. Maybe let's just switch gears a little bit. I haven't.
So, Eric, sorry for interrupting. You still haven't shown the poll on federated learning.
Yeah, no, I haven't. I could. I'm happy to run it. Let me write it up. But before I do that, let maybe try to. I'll introduce the sort of next topic, and just trying to do a few things at the same time here. So I think as we've seen, that word of AI is in a lot of minds. Certainly, we know from various industry reports that there are many efforts that are devoted with various levels of some of it being blue sky, some of it being a little bit more applied, to looking at what AI technologies can do in the drug discovery space. And I certainly would like to include that as part of our discussions. And we certainly have a pretty interesting approach in terms of what we've done with our AIDD module, which Michael described in his presentation today. And what is really pretty unique about it is that we support the use of mechanistic models, which we've presented before, as HTPK, and we've done a lot of development in HTPK itself. So the ability to use these kinds of models, which really have never been really fast enough to be considered as part of this kind of optimization loop at a fairly large scale. Now we know that it's fast enough. We not only do we know that it's possible, but it works today. So we and anyone who has that module can incorporate this module in HTPK can essentially couple the two. So maybe that's I'd like to maybe steer the discussion a little bit towards pushing the boundaries there, because there was a performance boundary that we pushed for being able to enable this. Part of it was with the parallelization of the code, which I think Marv played a very large part in, and that works for the standard properties inanimate predictor, but also for HTPK. So maybe we can cover a little bit of ground there, give a little bit of details about what we've done, and expand a bit on what Michael presented today. Who wants to start? David, you want to maybe start to give us a bit of a sort of foundation from your standpoint, and then maybe distribute it to contributors of the group?
Yeah, sure. So we have a couple of topics on the table here. Maybe I'll just, I'm maybe the least expert in AI here, but I'll at least set the table a bit, which is to say that, obviously there's a lot of interest in deep learning, so to speak, which is actually, perhaps a collection of different technologies. We did at some point, maybe in the past several years, put some effort into looking at whether some of the currently popular deep learning approaches would be beneficial for us in the development of our predictive models, the things we've been talking about, PKA and whatnot. I'm not completely familiar with the work that we did, but I think the bottom line conclusion for us was that on the data sets that we generally are using for the development of our models, that these more sophisticated deep learning approaches were not that beneficial relative to what we're doing now, which is more shallow neural networks, more traditional neural networks. So that is most likely a reflection of the fact that we're using relatively small data sets. We've talked a lot about the difficulty of getting access to high quality data, and for many of the properties that we model, we're talking about maybe hundreds to at most 1,000 compounds. That may not be true for the PKA model, which I know we've spent some time discussing, but for the majority of the models, unfortunately, we're quite limited in the amount of data that we can model. And so, the more sophisticated AI approaches may not be that helpful in that situation. We didn't spend an inordinate amount of time, I think, in that research, and there's interest in carrying that on. And that's, so far the work we've done is limited to predictive models. And then I think more recently there's interest in the use of AI for discovering new compounds. And that's an area, where I think we will also devote some effort. I know we've had some discussion about it. And I'm afraid that this is the point where, to further that discussion, I'm going to have to pass the baton off to someone else. But hopefully that at least gave a little bit of background about what we've done to date in terms of deep learning. But hopefully someone on the team will maybe provide some further thought on this.
So I don't know who's...
I could just kind of give a historical perspective of just computational chemistry in general. I've been, a computational chemist since 1991. I've worked for software firms, but when I came on the scene in the early 90s, there were very few crystal structures. The company I worked for kind of focused on ligand-based design. So we were the small molecule company, where there were a couple other software companies that were more focusing on protein structures, protein folding, docking, and whatnot. And of course, as the 90s proceeded, then you get more and more crystal structures. More and more people were interested in the protein structures and docking into there. And it was a bit more glamorous. You see a protein rotating around. It's a lot more interesting than a small molecule. And so it impressed people. And back then, computational chemistry software was very expensive. It could be $1 million, and computers were also very expensive with the graphics. But as the 90s moved on, people and I think this is true, we ended up looking back, overhyped computational models, and they didn't in the mid 90s, I guess. They were expecting a lot more, to go from discovery to clinical trial very quickly. And then, of course, you also had things like the Human Genomic Project, which we were going to solve all the genes, and we eventually did that. And people thought that would be the solve all our problems so that we could discover drugs for every disease. Then came combinatorial chemistry and high throughput screening, and people thought that would solve all the problems. And here we're 15, 20 years later, and we still have problems. And I think I think some of the overhype in the 90s made there was a little bit of mistrust in computational methods. And one of the things I see with these artificial intelligence is that it's good that we're getting more and more people involved with artificial intelligent drug discovery, but some of the basic tenets, like you can't have a triple bond in a six-membered ring, things like that. So people were making, compounds maybe that they were predicted were very accurate or are very active, but they weren't possible to make. And I think that's one thing our AIDD software utilizes, is that we try and make compounds that can be synthesized and are realistic. With the skepticism with computational chemistry, I think it's always been, is that the computational chemists come up with a compound that just is not synthesizable. And I think that was a stumbling block. And then when Marvin implemented this synthetic feasibility, it was really enabled our AIDD technology to make a step forward. So kind of babbled on, probably wasn't too coherent, but I'll let someone else talk to.
No, but I mean, it's fair to say it's gone a long way since the days of, I remember some of the early days of de novo design, and I mean, clearly some of the compounds that were coming out of these programs were, questionable to say the least. I think now we have a lot more flexibility to encode, transforms, chemistry transforms that have real, feasibility in the lab, as opposed to purely virtual structures that will probably never happen. So yeah, it's gone a very long way. There are other, I don't know if there's other people who wanted to contribute.
Yes, let me plug in something that we're actually doing right now. We try to answer the question that essentially every modeler asks themselves, or they should ask themselves, namely, are our current modeling methods optimal? How do they compare to whatever the world population of scientists is doing? Are they having something better? Are they doing it better than us? Are they doing the worse than us? If there are better methods, maybe we should investigate them, right? I'm sure there are some modelers in the audience. Some may use our ADMET modeler platform. Some may use some other platforms. But I think the answering these questions is very important on everybody's mind. And the way to get the answers is to host the challenge. And that's what we have been doing for the past 1 month and a half. The challenge is hosted on this website. I'm going to put the URL in the chat. Hopefully everybody will receive it. We are hosting a challenge to predict a blinded biological property from molecular structures. So we took one of the data sets that we have access to, and we blinded it. Okay, this is a property that is not disclosed. The descriptors are blinded. The endpoints are blinded. So to ensure that the comparison of various methods is fair. This website is essentially the member of, I think, the École Normale in Paris. That's who sponsors it. Please read the description. I highly encourage everybody to participate. There's no charge to participate. Once you register, you get access to the training and test data, and you can compare yourself against the others. Here's recent statistics. We have 98 individual submissions, 19 team submissions, and there are 34 competing teams. They can see the performance of their models on in this public ranking table. This is the state of February 14th. Here's a ranking of various submissions. There's a private ranking that will be used to actually pick the actual challenge winner. Is not visible to participants, but is visible to the hosting, the host, us namely, and the co-normal scientists. And this is great to compare your methodology against the world. I can hardly wait until the challenge ends. The challenge ends at the end of this year, so there's plenty of time to compete. That's all from me.
Yeah, Robert, I'll just mention that link you provided points to a login. Is there another link that?
Yes, you have to register. You have to register first. Once you log in, it will open the challenge for you. There are actually quite a few challenges right now hosted on this website. You can take a look, but none of these actually pertains to computational chemistry. Okay? You have climate forecasts and stock market predictions and financial applications. We are the only ones that actually are.
If you click on that link, where does that take you?
Yeah, this one. Yeah.
Okay.
But I have already logged in. Okay, so
okay, yeah.
All you have to do is register if you want to see the data and see the results of the challenge and so on. Okay? That's it. We do not participate, of course. That would be silly. We let the whole world have the edit and submit the predictions and so on. No. That's all.
So it's only really about. I mean, it's the regression method itself that is competing here. So the X
because
block
everything is blinded. Yeah, sure. Everything is blinded. So it's to ensure. We don't believe that people will cheat, but some may be tempted to knowing, for example, what a property is. Okay, some may be tempted to get additional data. Okay, to get ahead of others. Okay, so we want to keep it fair. Everybody shares the same data, the same descriptors, and this is really a competition between methodologies.
Yeah, no, very good. Great stuff. Thank you, Robert, for sharing this. I'm by popular request, I will launch the last poll on the level of interest in federated learning, and I think it's out. So let us know what you think, and I'm going to leave it up for about 30 seconds, as I've done before. And then I think after that we'll probably, if there's any last-minute thoughts, share those and wrap up the session within the next few minutes. Okay, we'll now end the poll. It's it's about half of the audience is is high and half is medium. So there's definitely some interest in it. I mean, it's maybe an avenue that holds a good bit of promise just because of the way it's done. So yeah, I think there needs to be there has to be some novel mechanisms to facilitate this sort of enrichment of the data. All right. Well, I think we have 4 minutes left in the session. Are there other closing thoughts? Let's just maybe go around the table for any closing thoughts. David, do you have any particular closing thoughts you'd like to share?
Not anything too profound. I think it was an interesting discussion. One area we didn't touch on at all was sort of advancing in the sense of supporting different deployment methods that are becoming more and more popular with our clients. So if you listened into the earlier talks, we talked about a new API option that we're adding. We have more and more customers who are developing pretty sophisticated infrastructures and are wanting to use our predictions in different kinds of ways within those infrastructures. So that's something that we're really trying to do. Everyone knows this is a company devoted to good science, but we also want to have good software and keep up with some of the modern trends. So I'm proud of the work we've done there, and I know we'll continue to try to support our customers in every way that we can.
Yeah. Well, thank you. Yeah, no, the API is a great thing, and I think our customers already know, even if they don't license the API, they already have 3 mechanisms, actually 4 mechanisms by which they can deploy Admit Predictor through the UI, through the command line, through the pipeline pilot component, and the NIME node. So that's already 4. The API is a 5th different way of deploying with the performance advantages that you mentioned. So yeah, that's quite nice to have really all of these choices. Any other closing thoughts?
No, I just want to thank everybody for attending.
Yeah.
We appreciate your time and support of our software.
Yeah, I'm going to extend a very warm thank you to all of you who participated, who attended all of these sessions this morning. Again, thank you for joining us in this session and the other sessions as well. We very much look forward to tomorrow. Please join us again for some different topics. And again, we appreciate you joining us. Have a wonderful rest of the day. And I'd like to thank also all of the speakers this morning and the contributions to the discussion panel. So a warm thank you to, again, all of the speakers for this morning's session. Thank you so much. And I will now stop the broadcast. Thank you again.
Take care, everyone.
Thank you. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye-bye.